end. But otherwise, thank you so much for joining. We're going to have Dr. Krishnamurthy share her screen shortly, but I just wanted to start by acknowledging CADASIL Awareness Day yesterday. We really just express our gratitude with how many webinars and educational opportunities have been presented so that we can all better learn and educate our community and hopefully advance the science. And that really could not be done without optimizing diagnostic and predictive tools, which really is the specialty for Dr. Krishnamurthy. So we are so grateful to have your expertise from Emory University and your work with MRIs and developing tools for better prediction and diagnostic clinical care. Thank you so much for presenting with us today. Just a final note for those who joined in, because this video is being recorded, your visibility here, if you prefer, you can exit the meeting and we'll send you a recording. But otherwise, if you wish to ask questions, they can be done privately and we'll just review those anonymously at the end. So thank you, Dr. Krishnamurthy. And when you're ready, you can share your screen.
Well, it is such a pleasure to be here. Thanks for inviting me. Let me share my screen. Okay, do you see the full screen version?
It looks good from my end.
Okay. Great. All right. Well, this was a small study that we did that tried to look at what does exercise do to our brain. So the title is Impaired Brain Lactate Transport in Older Adults Reduces the Benefits of Exercise. I'm a principal investigator at the Atlanta VA, as well as an assistant professor in the Department of Radiology and Imaging Sciences at Emory University. And it is great to have you all join me on this talk. All right. So we know that exercise is really good for the body, right? It helps your muscles, your lungs, your heart, but it also helps the brain. Like it boosts creativity, it enhances certain cognitive skills like memory and attention and concentration. It reduces stress and anxiety, and it's even thought to slow down the onset of neurodegenerative diseases, right? But we really do not know why. And so there's a few different hypotheses out there as to how exercise supports brain health. One of those is that exercise increases cerebral blood flow, so the blood flow to the brain, and the related nutrients that come with that, like the increased amount of brain fuel in the form of blood glucose and oxygen, right? And so how does it do that? Well, when we exercise, we increase blood lactate and blood carbon dioxide, and that actually increases the amount of acidity that comes to the tissue. So when we increase the blood, or when we increase the pH of the surrounding tissue, we actually make it so that calcium channels are blocked. And when those calcium channels are blocked, the muscle fibers that surround our vessels go from contracted to relaxed. So if you think about it, instead of being really, really tight and really, really small, we now relax those muscles that are around those vessels, and then those blood vessels open up, causing vasodilation, and that creates an increase in cerebral blood flow. So that's one possible reason of how exercise improves brain health. Another line of thought is that there's something called brain-derived neurotrophic factor, or BDNF, that also increases with exercise. Now, BDNF is important because it actually helps during the rewiring of your brain. And that doesn't happen just when we're babies and kids growing up. That happens throughout our life, right? BDNF can help in the growth of neuronal fibers. There's something called dendrites that can be activated with BDNF. And then there's also synapse and synaptic plasticity that's activated with BDNF. That's probably a little bit more long term. So what I'd like to talk about is another potential mechanism, which is exercise related changes in brain metabolism. All right, so let's think about what happens when there's a single bout of intense exercise. If we think about it from what happens on the periphery, like what happens in our muscles. When we first start the exercise, it's usually pretty easy, right? And at that point, the muscles are using all of your reserve glucose, so the sugar in your blood, as well as you're breathing normally, right? So you've probably got a lot of oxygen that's in your blood. And so that glucose is combusted with oxygen. It gets dumped into the TCA cycle, the electron transport chain, and it generates a lot of energy, right, in the form of ATP. And that muscle can use that very, very rapidly. Now, if you increase the workload, right, you make it a little bit harder, like you increase the gear or you start to sprint. At that point, you're increasing the workload and you're going to feel like you're breathing a little bit harder. Your heart rate is going to go up. Also, the gases that are exchanged in your lungs is going to change, right? A little bit more CO2 is going to come out, and you're going to be breathing deeper. You're also going to increase the blood flow to your muscles. And so with all of that, the muscles really can't keep up. And so it's going to switch from that oxygen-rich aerobic exercise condition to an anaerobic exercise condition. And that's really when glucose is going to undergo glycolysis. It's going to generate a little bit of ATP, and then lactic acid becomes a byproduct. Lactic acid gets turned into lactate, and then it doesn't just stay in the muscle, right? Even though sometimes we feel a little bit of a muscle cramp, it doesn't stay there. It actually gets washed away by the blood. And what it's going to do is it's going to increase the lactate concentration within the blood, and we can actually measure that. And so that's what happens in your muscles when you start to intensely exercise. Now, how do we derive energy from lactate? So first, here's a molecule of lactic acid. And if a proton dissociates from lactic acid, it now is blood lactate. So that blood lactate can be used in two different ways. First way is something called the Cori cycle, where that lactate gets taken back up by the liver. It's transported back into the liver and then converted back into glucose via gluconeogenesis, right? And so it's stored energy, which is really helpful. Another way that blood lactate can be used is lactate is transported to all other cells. So the heart, lungs, and yes, even the brain, where it can be converted back into something called pyruvate and entered back into the TCA cycle, where it can again generate a lot of energy for you. That hypothesis is called the lactate shuttle. So that's the way that we think the brain is deriving energy from lactate, even in the context of exercise. So let's think about brain metabolism. So we're used to, okay, glucose exits the blood, enters the brain parenchyma, then in the presence of oxygen, it enters the mitochondria. You get a lot of energy out, right? Just like most of our other cells in our body. We just talked about that in the context of muscles. Same thing happens in the brain. Now, if oxygen is not available, just like in the rest of our body, glucose gets turned into pyruvate, a little bit of energy comes out, lactate is the byproduct, and then it gets dumped back out into the bloodstream. So very similar processes happening across our entire body. Now, when we're exercising, actually a lot of that blood sugar is going to go to our muscles. So what does the brain do? Now, the muscles are using all of our glucose, but all that extra lactate was just generated. So what happens is that lactate actually exits the bloodstream, enters the brain, and then gets turned into pyruvate, and then can again get dumped into the TCA cycle to generate a lot of energy. So there's all these different pathways for the brain to make sure that it continues to work. Now, is this even possible? Yes, the answer is yes. So usually we're used to glucose entering the brain and then being metabolized, but there is this thing called lactate dehydrogenase, and there's a form A, which turns pyruvate into lactate, where it can then get dumped out into the bloodstream. But there's the reverse possible too. There's the form B, right, which is down here. So lactate can actually exit the bloodstream and then get turned back into pyruvate. And these are basically both forms, isoforms, are available in the brain. And so this has been shown. So hopefully by now I have convinced you that excess lactate generated from muscles during exercise can enter the brain via the bloodstream, right? So glucose generated in the muscle is then, or glucose broken down in the muscle can be turned into lactate. It then increases the amount of lactate in the blood, where it can then be transported into the brain and used as a fuel source. Now let's talk about used as a fuel source. So transporting lactate into the brain is actually not an easy thing. There's something called the blood-brain barrier, which makes it so that not a lot of stuff can enter the brain unless there's a specific transporter for it. So there is a specific transporter for lactate. It's this MCT, monocarboxylate transporter. There's different forms. There's forms 1, 2, and 4 that are actually found in the brain. So there's different forms, like for example, MCT3 is in the eye, in the retina. So MCT1 takes you from the blood into the brain parenchyma, right? So there's just one isoform to cross from the blood into the brain. The second isoform takes you from the brain parenchyma into the neuron. And then there's this MCT1 and 4, which can take you from the brain parenchyma into the astrocyte. So these are all different cells. The neurons are the ones that help us, send signals. Astrocytes are a type of cell that supports neuronal function, but it is extremely important. And we'll get into how these might affect things in just a bit. So if we then look a little bit closer at the neuron, because the neuron is really the source of signaling, right? So if we take that lactate and then we send it into the neuron, inside the neuron, we're going to have some metabolism. The neuron constantly requires energy, right? So usually we think of glucose turns into pyruvate, TCA cycle, but we can also have lactate turned into pyruvate and then into the TCA cycle. So when we think about TCA cycle and brain metabolism, we are usually thinking about, okay, let's get some energy in there to keep things moving, to keep things going, right? But in the neuron, there's this extra special, what we call a GABA shunt. So there's different molecules within this TCA cycle. So there's alpha-ketoglutarate. Alpha-ketoglutarate, you can actually shunt those molecules and you can generate a neurotransmitter called glutamate. Glutamate is the brain's primary excitatory neurotransmitter. It's going to make things go, right? So if you package that into vesicles, you now have the neurotransmitter glutamate, and that's what happens in a glutamatergic neuron. So a neuron that is responsible for making things go. Within that same shunt, if it's not a glutamatergic neuron, if you have an inhibitory neuron, meaning it's mainly going to be GABAergic, you can generate the molecule GABA. GABA is the brain's primary inhibitory neurotransmitter, meaning it's going to put the brakes on things. And so there's this constant push-pull during brain activity that helps some things go and some things get stopped. But it's a very healthy process of how the brain functions. So GABAergic, so if GABA is generated, packaged into vesicles, you now have the neurotransmitter GABA, and that's what happens in a GABAergic neuron. If neither of these are needed, that GABA can be remetabolized and put back into the TCA cycle for more energy. And in fact, there are studies out there, mainly from the Coxon group, that show that changes in blood lactate during exercise relates to changes in brain GABA. Now, this has only been shown in young, healthy adults. And with the study I'm about to show you, we're going to extend this knowledge to healthy older adults and determine if brain lactate is the mediator of that relationship. Another form of evidence that shows that exercise is important for brain health. If you are more fit, you actually tend to have higher concentrations of both GABA and glutamate and glutamine. So both your excitatory side of things as well as the inhibitory side of things have more stuff available to it if you are more fit. And this is true for both healthy and young, healthy older and younger adults. Now, why do we care about GABA and glutamate? This sounds so foreign, right? This is so abstract. So GABA is the primary inhibitory neurotransmitter and glutamate is the primary excitatory neurotransmitter. We talked about pushing the gas and pushing the brakes, right? So GABA is the brakes, glutamate is the gas. They both play key roles in cognitive functions, like strengthening neural pathways and encoding new information. So in lay terms, talking about learning something or relearning something, also memory, right? Capturing something and then keeping it. And both GABA and glutamate do this via neuroplastic principles called long-term depression and long-term potentiation. So LTD and LTP. And all of these are important principles in rehabilitation sciences. And so the overall goal of this study is to understand the factors that modify rehabilitation potential of an acute bout of intense exercise. So that exercise, what's going on in the muscle, how that then affects the lungs, heart, and circulation, how that then changes things in the brain, and eventually where we want to get to is then how does that improve brain function? And the hypothesis is that exercise-induced changes in blood and brain lactate are going to be associated with exercise-related alterations in neurotransmitter concentration, and that these relationships are going to extend to older, healthy adults. So the way that we tested this is we recruited both healthy, younger, and older adults, and we put them in the in a magnetic resonance scanner or an MRI scanner, and we collected brain GABA and brain lactate at baseline. We pulled them out, we put them on an exercise bike for 20 minutes, and that during that 20 minutes, it was a high intensity interval training. So they were basically cruising for a little bit and then asked to sprint for about 30 to 60 seconds, and then again cruise, and then sprint again, and then cruise and sprint again. And so we would do this in about 2 to 3 minute intervals, hence the high intensity interval training. And that really gets people's heart rate up. And so when you get people's heart rate up, you also increase their blood lactate. And so during this time, we also measured their blood lactate. The way we did that is with a little finger prick. It's almost like a glucose meter, but instead of sampling your blood glucose, we are sampling the blood lactate. Now, immediately after that 20 minutes of high intensity interval training, we put participants right back into that MRI scanner. And again, we collected their brain GABA and brain lactate. And this work was funded by the VA in a little pilot study. And so once we combed through all the data, made sure whose data was good and not good, we ended up with 5 youngers and 7 healthy older adults. Now, by definition, their age were different, right? So we had on average 25-year-olds versus 67-year-olds, but everything else was just about the same, similar across these two cohorts. Their sex distribution was very similar. Their MoCA scores, so MoCA is a way of looking at cognitive deficits. Their MoCA scores were very similar. Their years of education were very similar, as well as their body mass index, right? So how big were they? They were actually relatively similar. And finally, their activity scores and their sedentary behavior was actually very similar. So in a way, we did a pretty good job when we did the recruitment to make sure that the differences that we do detect is not going to be because of brain differences or because of body differences or activity differences. Now, as far as the 20 minutes of exercise is concerned, again, we tried to make it actually quite similar across the youngers and olders. And when we finally got a chance to look at the data, once everything was complete, the percent time spent in heart rate zones 1 through 5 was not different across groups. So we did a really good job there. We tried to actually keep them in zone 4, right? Because that's kind of where you're at the 75 to 80% in your heart rate zone. We also looked at their perceived exertion, and that was not different across groups. So from their perspective, from a participant's perspective, they worked very hard, but we didn't work the youngers or the olders any harder compared to each other. And then finally their heart rate. There was actually no group differences in the heart rate between youngers and olders. Now there was a group by time interaction, but that is not going to affect our data as is. So basically, whatever differences we do observe, it's not going to be because there were brain differences at baseline. It's not because there were any body differences at baseline, and it's not because we exercise them differently during the 20 minutes of HIIT. So similar exercise intensities. Now here's a picture of that blood lactate meter I was just talking to you about. So yeah, so we prick it with a little finger prick, and then we are able to measure the blood lactate with a little meter. And as the heart rate increases, the blood lactate also exponentially increases. It's actually quite interesting. Now, when we looked at differences in blood lactate that's generated, remember, we didn't work them any different. So the intensity of the exercise was actually quite similar across youngers and olders, but physiologically, they do end up reacting a little bit differently. At baseline, their blood lactate levels are the same. But after about 12 minutes of this intense exercise, the youngers really rise in blood lactate more than the olders. So both groups do cross that anaerobic exercise threshold. So they both cross that 4 millimolars per liter threshold. But the youngers really outdo themselves versus the olders. They're going above that anaerobic threshold, but they are not rising as quickly as the youngers. Now, for the brain metabolites, we use something called magnetic resonance spectroscopy to non-invasively measure brain metabolite concentrations. And we did that at 3 Tesla with edited megapress. Now, here is a raw lactate MRS signal. No, there's no audio on this on the video. So there should not be any audio. And the videos that I'm sharing, it's only like PowerPoint animation. Okay. So let's see. So this is a raw lactate MRS difference signal. It looks very noisy, and it is. But remember, we're doing this non-invasively, so we're not having to cut anyone open to do this. Luckily, we are able to model the signal very nicely. And if we look at the LC model basis sets, you can see that the lactate is over here. So in our whole spectrum, we should be looking for a signature over here. And indeed, we see that. So this is our brain lactate. And if we look at the younger versus the older, you can see from pre in the red to the post in the blue, the youngers rise in brain lactate, while the olders seem to reduce. And you can see this happens for each and every participant. And I've split them up into older and younger. So each of the youngers rise, and in the olders, each of the olders, except for one, they reduce. This was very unexpected. I thought that everyone was going to go up, right? Because their blood lactate went up. So I'm thinking, hey, if their blood lactate is going up, their brain lactate is going to go up too. That seems to be only happening in the younger. So it's going on in the older. Remember, at baseline, they're not different, right? Brain-wise, they're the same. Body-wise, they're the same. Exercise, intensity-wise, they were the same. So what's going on? We did have to think about, well, tissue averaging. So inside this MR spectroscopy voxel, you can see here it's quite large. So there's a lot of tissue that gets dumped into this thing. And one of those tissue types that gets dumped in is the blood lactate. And so we had to ensure that there was no cross-contamination between the two numbers. And so we did some correction. But in the end, even after correction, we have the same story. The youngers go up while the olders go down. And so we really, now that we have the blood lactate and the brain lactate, now we can try to understand what is governing neurotransmitter change. So to look at the brain neurotransmitters, we collected a GABA MRS. So similar sort of acquisition, but now tuned to brain GABA and brain glutamate and glutamine. This is what the spectrum looks like. And here is glutamate and glutamine on this side, and here is GABA. And so the signatures that we want to look for are here and here. And there you can see there's not much covering them. So we are able to actually extract those with quite a bit of confidence. And when we look at exercise-related neurotransmitter changes, we can see that the older's brain GABA significantly goes up versus the younger brain glutamate significantly goes up. And so, again, we have a little bit of a mystery. Why is it that exercise causes GABA to change in older and glutamate to change in younger? For that, we have to go into the literature and look at the energetic cost of GABA and glutamate cycling. It turns out that the amount of energy required to cycle GABA is 20% less than the energy required to cycle glutamate, right? So it's actually energetically cheaper to cycle GABA or to change GABA than it is to change glutamate. So perhaps it is cheaper for the older participants to increase GABA, while younger participants can afford to increase glutamate and glutamine. Again, this is speculation, but it is very well backed up by the literature that this is what might be happening. So now that we have all of these things in front of us, we can try to understand, is it the amount of blood lactate during exercise that sort of relates to how much the neurotransmitters change, or the reserve brain lactate, right? And so when we do the statistics, we see that it is indeed the amount of blood lactate rise that sort of gives the fodder to change the amount of brain GABA. When we do this with glutamate and glutamine, there is no relationship. Again, unexpected, but I think I have an explanation. So complicated diagram, and I apologize, but this is probably one of the more simplified ways of displaying this. But let me take you through it. It's something that we've already seen. So here is the blood, and we have glucose and or lactate entering the brain parenchyma, and this is the neural side of the equation. So again, we're going to go through our energy cycle, and we can shunt alpha ketoglutarate into glutamate or GABA, right? So this we've already seen. Now there's another way. There's remember we talked about that lactate can also enter astrocytes. So astrocytes are supporting cells. They're not the ones that are actually going to create the firing or the signaling. The astrocytes support neuronal signaling. So again, here's the blood side of things. The glucose and lactate can enter the brain parenchyma and be metabolized within the astrocyte. Again, there is a GABA shunt within the astrocyte. However, it is only towards glutamate. There is no what is called GAD65 or GAD67 within the astrocyte. So glutamate will never be generated into GABA within the astrocyte. However, glutamate can be packaged into glutamine, sent back into the neuron, where it can be, again, repackaged into glutamate. So there is this very fancy cycling of glutamate and glutamine that kind of gets shared between the neurons and astrocytes that does not happen with GABA. So that's one thing that is interesting to note. Now, when GABA gets remetabolized into the TCA cycle, whether it's on the neuronal side or the astrocyte side, it disappears. It's no longer GABA. We're not going to detect it. Now let's try to see where we are detecting the GABA. So we talked about that GABA, if it's shunted into the GABA, or the molecules, if they're shunted into the GABA shunt and it's a GABAergic neuron, it can be packaged into vesicles. There, it's now ready to be a neurotransmitter, right? So if it's signaled that, hey, release GABA, there's something called exocytosis, where it's released into the synaptic cleft, and there it has multiple fates. It can either do its thing and attach to a GABAergic receptor, right? And so now it has done exactly what it was meant to do, which is signal the next neuron over. The other thing that can happen is that GABA can be retaken up by the astroglia and remetabolized, or GABA can also spill over into the outside, where it becomes tonic inhibitory tone. Now, let's see which ones can we actually detect. The first two, I'm going to say, are MR invisible because they're tightly packed. It's a short T2. We just don't detect it. Same thing in the synaptic cleft. It's a narrow gap, short T2. We really don't detect it. When it's attached to a receptor, it's attached to a large protein. Again, a short T2, we cannot detect it. However, it is that spillover that we are probably detecting. So that increase in inhibitory tone is what we are sensitive to. And basically, this means that, hey, the neurons are firing more. There's more chance of spillover. And the entire the entire the entire system is basically on overdrive. Now, why are we not seeing the same sort of relationships with glutamate and glutamine? Probably because there's multiple pathways to generate glutamate and glutamine, not just through metabolism, but also within this cycling between the astrocyte and the neurons, right? So it doesn't necessarily have to be directly hooked into the metabolic process. So why would that brain lactate decrease in older individuals, even though blood lactate increases? That's a question that we just posed, and we haven't answered quite yet. So one thing that happens is that cerebral blood flow and something called cerebrovascular reactivity, they decrease with age. So we talked about blood flowing through those vessels. That tends to decrease with age. And then also the ability to dilate the vessels that supports that blood flow also decreases with age, likely because the vessels are hardening and all those things. Now, one thing that happened with blood flow and vascular activity is that these are pH dependent, pH dependent, and they're regulated by pH sort of activities, right? So but there's a double whammy with these pH dependent issues. The transport of lactate across the blood-brain barrier is also pH dependent, right? And so now you have two aspects of brain lactate, of lactate transport that are reduced with age. So even though you have a reduction, so you have this reduced blood lactate, which reduces the pH increase. And that means that you're not going to have as intensive a vasodilatory sort of response. So a reduced pH related vasodilation means a reduced arrival of lactate. But even if lactate arrives, a reduced pH related lactate transport equals a reduced transport of lactate into the brain. So you have this cascade of, well, you're not increasing pH as much, which means you're not going to dilate it as much, which means you're not going to bring as much lactate there. And even if you could, you're not able to transport the lactate across the blood-brain barrier. And then the final double whammy is, even if lactate can enter the brain, you have this ratio of A to B in that lactate dehydrogenase sort of shifts with age. So in younger age, lactate turning into pyruvate is favored. So this pathway is favored, versus when we're older, pyruvate turning into lactate is favored. So there's all of these barriers, and as we get older, I don't know, things just change, but we need to figure out how can we come back to these, to reestablishing a healthy brain metabolism, even in older age, so that we can either reacquire lost skills or maintain function for as long as possible. So just as a recap, I hope I've convinced you that both younger and older participants can participate in high intensity interval training. In our case, we were able to make the exercise session equally intense across both groups. Now, even though it was equally intense, blood lactate did rise more in the younger compared to the olders. And after correcting for blood lactate, brain lactate still increases in youngers and decreases in olders. So what that meant for us is that brain lactate is metabolized in older adults above and beyond that increased supply in blood lactate. And perhaps the lactate in the blood is not able to be transported into the brain. And then the blood lactate in younger adults is transported into the brain to be metabolized. So that their things are working the way they are intended to. And that supports uh, the way, yeah. And then finally, brain GABA increase is related to blood lactate, but not brain lactate, which supports the lactate shuttle hypothesis. And glutamate and glutamine increase is not related to either blood or brain lactate pools, likely because there's multiple pathways for that generation. And so that kind of wraps up my slide deck. And I'd really like to thank you for your attention, as well as my multiple collaborators, especially Giri Krishnamurthy for his insight in brain metabolism, the exercise core for their help in establishing the exercise protocol, Anastasia Bosali for her help in processing the spectroscopy, Mohammed Saleh for his insight on the brain lactate acquisition, and Steve Wolf on rehabilitation sort of aspects. And I'd love to take any questions that you might have.
Thank you so much. This is such a timely kind of topic with longevity science, and aging science is really kind of a lot of what we're hearing about these days. So I just love that it's being studied so properly and precisely to really have quality information. Thank you for that awesome presentation. I am going to read, we have 2 questions for you.
Okay.
For context, the person is an exerciser, and they are asking if there are any type of supplements that they can take to help with this. This, I'm assuming they mean the reduction in lactate transport, or anything else that they can do as an exerciser.
That is such a great question. And honestly, I do not have an answer for you. I wish I did. There's just so much more that we need to try and understand what is making this work. I mean, this is basically an unexpected result, and this is going to be probably scrutinized up and down when we do publish this, because it's going to be really hard for people to try and figure out, well, what do we do about this? And like you, and just like your question is, how do we change this? How do we attack this in such a way where we do make the metabolism work in such a way where, you know, even if we're exercising, it's not getting to the brain or the rest of the body in a way that it does when we're younger, right? So and that's frustrating. It's frustrating for me. So yes, great question, and I love that you're attacking this from a nutrition perspective, because that's the way to go.
Thank you very much. Our second question is possibly somewhat related, but maybe you can speak a bit to the blood-brain barrier. This is a term that we hear a lot about and not many of us fully understand. But the question relates to, similarly, is it worthwhile to take supplements and other things that we hear about improving aging and older adult health, or are these also overkill or not reaching the brain or doing things that we need them to do?
That's a great question. I mean, not a lot of things are going to cross the blood-brain barrier. Now, if you take something like there are actual GABA supplements that you can buy at the store that does not cross the blood-brain barrier, right? But that might actually get into something like your muscle. Your muscle can also use GABA. So a lot of the supplements that you take are not going to directly cross the blood-brain barrier, and therefore directly impact the way your brain metabolizes things. However, I encourage everyone to eat a healthy diet, because it's really when we have the right building blocks that our body has broken down naturally, like proteins get broken down into amino acids. Those amino acids are then used to generate things like GABA and everything within the brain, right? So you need those building blocks, and you get those through natural consumption of healthy, lean food, high in protein, but try to see if you can also get your proteins from lentils and beans, not just meat, you know. So yeah, it's supplements are good, but they're not always going to cross the blood-brain barrier.
Appreciate that. Thank you. We did have one additional question come in if you have time.
Sure.
It asks how these findings relate to CADASIL patients. That's kind of broad, but I guess I'm thinking specifically in terms of, you mentioned vascular changes as just in general for all older adults, but in terms of the vascular impact in CADASIL, how would you relate these findings?
That is such a great question. So, what I didn't show you today is in this little study that I did, we also did this in chronic stroke participants. And so we are starting to look at this in patients who have vascular disease, underlying vascular disease, that results in stroke and those kind of things. What we are finding is that we are able to still intensely exercise even patients with chronic stroke. As long as a doctor okays it, as long as your stress test is cleared, exercise is possible. Exercise is good for you. It is recommended by the American Heart Association and the American Stroke Association, so that you continue to move, you continue to keep your body going. Now, in the cohort of stroke patients that I did this intense exercise on, and again, I collected the brain GABA and brain lactate at baseline, and within and immediately after that 20 minutes of exercise, I saw that increase in brain GABA. Okay, so we can modify the neurotransmitters even in stroke patients with exercise. Now, the missing gap is, and I don't know, is how does this improve function? How does this improve learning? How does this improve memory? I don't know yet, but that is the immediate future work that we are working on right now. We are doing a 12-week intense exercise program in stroke patients, and we are seeing remarkable results, not just in their walking ability, not just in their mobility, and not just in their endurance, but also in their attention, in their verbal learning, in their visuospatial learning. So cognitively, they become better. They report to me that they feel so much better. They're able to do things again that they used to be able to do. And so I would love to take this from the stroke patient realm into CADASIL to see, hey, how can exercise at least preserve brain function, even when there is that looming neurodegenerative thing going on in CADASIL, right? So can we keep the brain going? And keep things from a metabolic perspective, keep things going so that you continue to feel good, so that you can continue to keep doing what you want to do. And so that's how this relates to CADASIL. It's a future sort of perspective, but that's where we're going.
Thank you so much. I think that answer also is really relatable to all adults as we age. And I think vascular health is also an issue for all of us as we age. So really relatable, and appreciate your expertise and your time. And thank you so much for being part of our CADASIL Consortium research project. We look forward to sharing future webinars and keeping in touch with the community. Thank you so much.
Thank you, everyone. It was a pleasure seeing you all.